Today I'm going to be teaching you how transcription works in a prokaryotic organism. We're going to be using these pieces of candy as nucleotides. Prokaryotic transcription is the process of forming messenger RNA from a DNA template strand. So we start the process of prokaryotic transcription with our RNA polymerase enzyme right here. Right now, this complex works, but it has no way of binding to the promoter region up here. In order for it to do that, it needs a sigma factor. A sigma factor is going to bind to this RNA polymerase and turn the core enzyme into a hollow enzyme. Enzyme is going to bind to the DNA template strand in the promoter region up here. Now in our promoter region, we have two consensus sequences. We have our -35 region and our -10 region. Our RNA polymerase is going to bind to this region, to both of these sites, because of its sigma factor. RNA polymerase is like a multi-tool. It has to do multiple different things for transcription to occur. So what it's going to do is it's going to travel along the DNA template strand. It's going to recruit RNTPs, or ribose nucleoside triphosphates. It's going to cleave two of their phosphates, and it's going to add them complementary to each nucleotide on the DNA template. Now, once it does that, it's going to join them using phosphodiester bonds, so that way the mRNA sticks together after it leaves, once the DNA is rewound back up, while also unwinding the DNA in the front, and it's also rewinding it in the back. Now that the sigma factor is gone, this is going to be reduced to a core enzyme rather than a hollow enzyme. So for row-independent termination, we're going to have what's called a palindrome sequence with the mRNA from the DNA template. So what's going to happen is this G and C rich area is going to bind to itself. So what follows is a loop in the GC rich regions that bind complementary to each other with A's and T's at the end that create this hairpin loop that is followed by A's in the DNA template strand and U's in the mRNA strand. These have very weak bonds, so it's going to dissociate. The RNA polymerase is going to fall off, and then this is going to detach. For row-dependent termination, we're going to have a C-rich binding site on the mRNA, and we're going to have a row factor, which is a helicase enzyme, bind to the C-rich site and slowly move up the mRNA to dissociate the RNA polymerase, the mRNA from the DNA template, and the row factor itself. Consensus sequences are the most commonly found nucleotide in a string of nucleotides at a specific point on the DNA. Let's say we were to have an insertion between the -35 and the -10 regions. Since there was a 5 base pair insertion at the point between the -35 and the -10 region, the -35 region is going to change because the optimal nucleotides have been shifted over. The sigma factor will have more trouble binding to this new region because of the new nucleotides.